I would like to welcome everyone for this last session of this day on celiac disease. So this last session will be on tolerance and celiac disease. So first of all, I would like to thank our sponsor for this session, the Celiac Disease Foundation. So we're going to start in a few seconds. I'm still going to wait a little bit because I think some participants are still logging in. I think we're good. Yeah, we're good to go. So again, welcome everyone. So our first speaker for this session will be Dr. Eric Palmer. So Dr. Palmer is a professor of medicine in the Department of Medicine, Microbiology and Pathology at the University of Chicago. He's the director of the Duchossois Family Institute that supports research linking the microbiome and diverse diseases. And today is going to talk about leveraging the microbiome to treat complex immune disorders. So welcome, Dr. Palmer. You can share your screen.
Thank you very much, Valerie. And let me see if I can bring this to slideshow mode. There we go. So yes, this is my disclosure slide. I will talk to you today about the microbiota, and I'll start by telling just a little bit about our institute, the Duchossois Family Institute, which is at the University of Chicago. And one of our main foci of investigation is the role of the microbiome in human health in general. And the last decade has really seen remarkable progress and remarkable discoveries on the impact of the microbiome on a variety of disease states that extend from the development of obesity to susceptibility to a wide range of infectious diseases, and of course inflammatory bowel disease as well. And I think for celiac syndrome, there is certainly no question that the microbiome is going to have or has an impact. Now, the idea that the microbiota or the microbiome can impact human health goes way back. And in fact, over 100 years ago, Ilya Mechnikov was already postulating that the bacteria that live in the intestine can enhance health. He was particularly fascinated by the ingestion of yogurt and longevity in Bulgaria. And back then, even over 100 years ago, was postulating that lactobacilli in the intestine can enhance health. And over 50 years ago, Rolf Raeder at Loyola University in Chicago was postulating that being able to control the microbiota, or what he referred to back then as the bacterial flora of patients, would be of great value in enhancing the health of our patients. Now, we have learned a lot about how the microbes that reside in the intestine impact the immune system that is just underneath the epithelial layer of the gut. And it has, with each discovery, become increasingly complex. But I think one way of thinking about what is going on with the bacteria that live within us in the gut is to think about how they interact with each other. They are, in fact, an ecosystem with hundreds of species residing together in a very tight space. And so they interact directly against each other. And they can, in fact, either support each other or inhibit each other. But there's a second really important way that bacteria interact with each other in the intestine, and that is to actually go indirectly by stimulating the host, which can drive the host to produce antimicrobial factors, for example, that can then inhibit competing bacteria. And more of these kinds of mechanisms of bacteria competing with each other are being uncovered. So what I'm going to do, there are so many different ways of actually starting to discuss the impact of the microbiome on human health. What I'm going to focus on is how dramatically healthcare that we provide to our patients can alter the microbiome, and the impact of those changes in the microbiome on longer-term health aspects of the patients. So what I'm showing in this plot, just to familiarize you with some of the ways that we show microbiome compositions, are the microbiomes of about 15 healthy donors that we sequenced. And each bar in this bar graph represents an individual donor, fecal sample of which was sequenced. And all the colors that are shown in this bar graph here represent different bacterial taxa or genera or species. And what we see in all of our healthy donors are fairly similar color patterns, although the proportions are somewhat different. And the aquamarine here are bacteria that are well known as inhabitants of the gut. They belong to the Bacteroidetes phylum. They're obligate gram-negative anaerobes. The pink are a family of bacteria that have, over the last decade, been discovered to produce many health-promoting factors, including short-chain fatty acids. These are the lactospiraceae. And then these green here are the Ruminococcus. And the pink and the greenish guys are composed of many different species, but they share some characteristics in that they are gram-positive, they're obligate anaerobes, and they produce a variety of factors that we are increasingly learning enhance our health and provide disease resistance. So probably for the last 10, 12 years prior to my arrival in Chicago a year ago, we were studying bone marrow transplant patients. And these are, for many reasons, very interesting patients to follow. But one thing we took advantage of is that they're in the hospital often for many weeks. And we were able to collect fecal samples from them and sequence them and see how bone marrow transplantation changes the microbes that reside within them. And of course, there are many risks associated with bone marrow transplantation and complications. And one of them is graft versus host disease, which is often an immunologic attack of the bowel mucosa. And so we wanted to know what happens to the microbiota. And what we see here is one patient, and this is a timeline here. And the patient came in with a lot of the bugs that we like to see, a lot of the pink and a lot of these sort of olive green. But as the patient went through the transplant, they lost all of the health-promoting bacteria, and they were replaced by bacteria that we in the clinic recognize as troublemakers. So here's Escherichia coli, turns out to be an antibiotic resistant one that ended up in the blood of the patient. And then that resulted in antibiotic changes. And then the patient was dominated by another troublemaker, Anaerococcus faecium, which is also highly antibiotic resistant. So these changes were really dramatic, associated with high infection rates. And over the last decade, we were able to characterize the microbiomes of about 1,400 patients. And we were able to sequence over 14,000 fecal samples, and we were able to plot them and kind of in a way define the universe of microbiome compositions that patients have. And we found regions as we plotted them that were associated with high rates of Anaerococcus infection because that was the predominant organism in their gut and other regions. What we also were able to determine is that this is a very good neighborhood to be in because that's the neighborhood where the microbial populations are the most diverse. We also wondered, of course, if beyond infection rates, if you lose your diversity of the microbes in your intestine, does that have longer term implications for the patients? So we ranked patients from those who maintained their diversity down to those who completely lost it. This is a 94 patient cohort. We followed them for 3 years to look at the impact of diversity loss at the time of bone marrow transplantation. And what we found was quite remarkable, and that was that if you lost diversity, your mortality rate over the 3 years following transplant was over 50%. But if you maintained it, your diversity, your mortality rate was under 10%. So that suggested, didn't prove, but it suggested that the microbiota can have a very big impact on outcomes after bone marrow transplantation. We recognized that most of the bacteria in the gut actually stay in the gut, which is good. We don't want them in our blood. But what they do is they produce metabolites. And it's in fact the metabolites that these organisms produce that are probably responsible for their biologic activity on the host. And we were lucky to be able to work with the metabolomics core at Memorial Sloan Kettering to characterize the the metabolites in the intestine. And what we found, this is another transplant patient who came in with diversity, lost it. But along with that, the beneficial short-chain fatty acids that immunologists have studied and have demonstrated impact, for example, T regulatory cell development, were well represented at the beginning, but were lost during the course of the transplant. Along similar lines, we've now learned more and more about how the normal bacteria that live in the gut modify bile acids to produce, for example, deoxycholic acid and lithocholic acid, which are present in the setting of a normal microbiota, but then are lost as we go down. So as you become more and more purple, it means that that these are underrepresented, and red is that they're highly represented. So we know that, at least in bone marrow transplantation, that many of the beneficial metabolites that are produced are lost. Now, since arriving at the University of Chicago, we started a number of different clinical studies. One is with the critical care unit. This is working with J.P. Kress and Matt Stutz and Bhakti Patel. And we've collected now or enrolled over 120 patients and characterized their microbiota. And I think the only message to get from here is that the work that we did with bone marrow transplant patients in New York is not unique to New York bone marrow transplant patients. And in fact, here, what we're seeing is mixed among all these patients here in the medical intensive care units are many who have completely lost their normal microbiota. So loss of all the pink and the aquamarine, and a domination by organisms very similar to what we saw in the bone marrow transplant patients. Here, we're just plotting the bacterial families that we are particularly fond of, Bacteroidetes, Lactobacillus, and Ruminococcus. And you can see, we'd like to see them in these sorts of proportions, but they're gone in many of these patients. And when we measure metabolites, which we do now in the Duchessois Metabolomics Core facility, we see that many of our patients, we're also looking at liver transplant and heart transplant patients, have completely lost propionate and butyrate concentrations. And we think that probably has implications for their immune system. So our goal, of course, is not only to sit and document changes in the microbiota in our patients, but to be able to intervene. Our strategy is to be able to identify patients who have adverse clinical outcomes, identify among the bugs that they are harboring, which ones are associated with bad outcomes and which are associated with good outcomes, and then to be able to alter the microbiota. And we're doing that by having established a biobank of commensal bacterial strains. We started this in New York, but we're continuing it here in Chicago, and we've got now close to actually 2,000 bacterial strains that have been whole genome sequenced and annotated and characterized. And we now can plot these as well according to their genomes and see that, of course, bacteria that belong to a particular species like anaerobic hadrus all cluster together, but with some diversity. Others like Ruminococcus gnavus fall into different clades because of genomic diversity that they have. We can now also characterize each of these individual bacterial strains and ask, for example, how well do they acidify, and also how well do they produce butyrate. And we can see that all the butyrate producers shown in red are regionally distinct from the ones that don't. All the acidifiers are down here and belong to the genus Blaudia. We also, when we start to look at Ruminococcus gnavus, for example, and we see these different clades within the species, and this one was particularly interesting because it has been associated, certain strains, with inflammatory bowel disease, that in fact it's only one of the clusters here that encodes the polysaccharide synthesis locus that's been associated with inflammatory bowel disease. But what we're doing in order to really characterize these strains in greater detail and to be able to put ourselves in the position to use them as biotherapeutic agents is looking broadly at their metabolite production and their metabolite utilization. And so here, going across the top, we are looking at 273 strains of Lactospiraceae. And so, for example, for Ruminococcus gnavus, we've got probably about 20 or so strains that have come from different donors. And we start to then look at the metabolites that they produce. Production is shown in green, and depletion is shown in this dark blue or purple. So the darker the green, the more is produced. And so here we see all the butyrate producers. So these are bugs you may never have heard of, but they're important. Coprococcus comas, anaerobic hadrus, Eubacterium rectale. Healthy, health-associated microbes that are big-time butyrate producers that are likely to impact immune development, in particular T regulatory cell development. So then we have others as well that are succinate producers, also associated with disease resistance. And then we can learn a lot about what they utilize. So here are many of them are big-time amino acid utilizers in order to ferment. But what this gives us the ability to do is to start to pick from these various species in order to combine them so that we don't have bugs that are going to be competing with each other, but rather bugs that are going to be supporting each other. So this is my summary slide. We are very interested in moving forward this concept of being able to reestablish a healthy microbiota in individuals or in individuals with different illnesses, to be able to supplement the microbiota with specific strains with known functions that have been well characterized. And the Lactospiraceae are our first ones, but we are doing similar work now with Bacteroidetes and Ruminococcus as well. So these are my colleagues that I work with at Memorial Sloan Kettering. Justin Cross did the metabolomics work, and he's been very helpful in, with us establishing a similar, in fact, identical metabolomics core at the University of Chicago. These are my bioinformatician colleagues, and these are the clinical colleagues now at the University of Chicago who are working with us to characterize the microbiota in the MICU in heart transplant and in liver transplant. So my watch tells me I'm at 3:05. So I believe I'm done. Thank you.
That's perfect. Thank you so much, Dr. Palmer, for this inspiring talk. So I think you can, yeah.
I stop share?
Yeah, perfect. Thank you. So our next speaker is Dr. Shavonsky, who is a professor of medicine and pathology at the University of Chicago. So Dr. Shavonsky has a long-standing interest in understanding the immune mechanism leading to autoimmunity, and in particular type 1 diabetes. And we're actually thrilled that he's now interested in celiac disease, and I'm looking forward to his talk, which is entitled The Role of Gluten in Type 1 Diabetes. You can go ahead, Sasha. Yeah, we don't see you, we don't hear you for now. We're only seeing your slide, but we're not hearing you. You can unmute yourself.
We still cannot hear you. Can you please stop sharing to turn on your video and audio?
Okay, now I hope you can hear me.
Great, thank you.
Yes, I was going to say that I want to thank the organizers. So today I'll talk about the microbial connection between celiac disease and type 1 diabetes, and there is a question mark because it's a very fresh study. It would be nice to have a financial interest, but I have none. And so the question which was discussed today already at length is the connection between celiac disease, which affects the intestines, and the type 1 diabetes, in which, as you can see, the target cell is beta cell inside the islet. And the beta cell antigens get into the lymph nodes where the T cells are activated and then go back to the islet and kill beta cells. So the question is, what are the connections between celiac disease and type 1 diabetes? Genetics, we discussed it already, and also environment. In environment, the two most strong features of environment are diet and microbiota. So a few years back, we wrote with my student, Lonya Yerkovetsky, we wrote this treatise for cell host and microbe, in which we discussed the possibility that diet can affect the target organs along with the microbiota, either directly or through microbiota. And the same can happen even during the embryonic development, during gestation, so that will leave the lifelong effects. So our initial question was, how did we get to this study? We wanted to know whether the hydrolyzed casein, which was shown by many labs before us to be protective for against type 1 diabetes in animals, in rodents, whether it's the protection that it offers for the animals, is it dependent on microbiota or not? And that was a very simple question, as we thought at the beginning. So we built this germ-free facility at the University of Chicago. Now everybody uses it. And we use these mice that have no microbes at all, germ-free, to ask whether this diet will work also in germ-free conditions when there is no microbes. And in fact, it did. So SPF is specific pathogen-free, the regular mouse in conventional facility. And you can see if we feed them with chow diet, they develop type 1 diabetes. If we feed them with hydrolyzed or even non-hydrolyzed casein, they don't. And the same happens in germ-free conditions. So the conclusion from that was that protective diets work in both germ-free and SPF conditions. So we got the answer. The hydrolyzed casein diet activity is microbe independent. So if it's microbe independent, we have to study the physiology of the workings of this diet. And it turns out, I'm of course jumping through the results, but the most important one is that the protective diet reduced secretion of insulin by beta cells in the animals. In this experiment, we injected, this is glucose tolerance test, you inject glucose into bloodstream of animals, and they clear it within a certain time, in 2 hours. And the mice on chow clear it very fast, whereas mice on protective diet clear it very slowly. And the reason for that is that mice on chow, as already pointed out, secrete a lot more insulin. That's why glucose is cleared much faster. So the high insulin secretion causes higher beta cell stress. We're not discussing details of that, but we assume that's what's happening. And the model explaining the contribution of damage done to insulin-producing cells by cell stress to the overall diabetogenic process is here. So during type 1 diabetes, you have the overall damage of the pancreatic beta cells that results into hyperglycemia and loss of insulin control, or glucose control. However, if you can lower the intrinsic damage done by genetics and environment, then you can lower the probability of hyperglycemia development. And that's what this fantastic diet does. So that was the case, but of course we wanted to spoil the good thing and see how resistant it is for other environmental influences. And of course, the easiest way to spoil a good thing is to use gluten. So we used the same diet, but we spiked it with 4% by weight of gluten. And here's the glutens of 2 different sources. They're commercial glutens made by giant companies. So we fed mice with chow, they developed diabetes. With protective diet, they did not. And then protective diet spoiled with gluten, again, mice developed diabetes. Gluten doesn't increase insulin secretion, so that wasn't the answer to the mechanism of how it would happen that gluten spoils the effect of hydrolyzed casein diet. It was not through regulation of insulin secretion. Then what? So we asked the question, how does gluten work? And we have 3 suppositions. One is that it potentially activates transglutaminase 2. It can produce biologically active peptides, and whether anything of that depends on the microbiota. So to answer the first question, we used what is now known as CRISPR-Cas9 method of knocking out genes. And in this diabetes-sensitive mice NOD, we knocked out transglutaminase 2 gene. So you see that the protein is absent, but there was no difference in diabetes development between mice that had transglutaminase and the mice that did not have transglutaminase. So that wasn't the answer. So in mouse diabetes, transglutaminase 2 doesn't seem to be involved. So then we had to go back to the germ-free conditions. And we did this experiment by feeding mice this protective diet in germ-free conditions. So as you can see in blue, this is the gluten-containing diet. And in germ-free conditions, it doesn't stimulate development of type 1 diabetes. It's still protective. So the only conclusion we could make from this experiment is that gluten promotes type 1 diabetes, and it does it in microbiota-dependent manner. Now, how? So this is a typical experiments of what we're doing. We're taking germ-free mice. We colonize them with microbiota from SPF mouse, and then we put them on 3 different diets. And later, we take their intestinal contents, and we isolate microbial DNA and sequence 16S RNA genes, which then tell us who is there. Eric just introduced you to this technique. So in the small intestine of this ex-germ-free mice, we can see that mice on hydrolyzed casein diet are very different from mice on hydrolyzed diet, casein diet plus 4% gluten. So gluten changes a lot of microbial composition. And of course, we're looking for microbes that can digest gluten, so to produce something that can affect the biology of the mouse. And so we turn to the bacteria that can digest gluten. So here you see a Petri plate, which is covered with agar, with gliadin in it. So gliadin is not very well soluble in the water, so it's milky. And when the microbial colony can digest gliadin, you see this halo of free space, which is product of digestion by the proteases secreted by bacteria. And here is an example of plate which was seeded with bacteria from a regular mouse from the colony. So there are bacteria that can digest gluten. Again, summarizing what I'm going to tell you is that digestion leads to two major effects. It is activation of adaptive immune system, T and B lymphocytes, and activation of innate immune system. So first, I'll tell you the story about adaptive immune system. We did experiment which I never believed could work, but it did. With the help of Ludvig Solid from Norway, who provided T cells that are specific to gliadin. We took bacterial culture, we added gluten to it, cultivated overnight, filtered supernatant, and then added to the supernatant, we added T cells and antigen-presenting cells. If T cell is activated, the peptide is present, then it will produce cytokine called interleukin-2. And here is the result. So if there is no gluten added to bacterial culture, there is no IL-2 production. But if we add gliadin or we add gluten, we see high levels of IL-2 produced. So that suggests or shows that digestion of gluten by a particular bacterium can lead to activation of specific T cells. And one of these bacteria is Enterococcus faecalis, a very common commensal bacteria, which could also be pathogenic. In the wild type bacteria, you can see these halos of digestion. And there is a mutant that lacks two proteases secreted proteases present in E. faecalis. And if we look at IL-2 secretion, then protease positive bacteria stimulate interleukin-2 production, and protease negative do not. So bacteria secrete proteases that liberate peptides, stimulating specific T cells. That is adaptive side of immunity. What about innate responses? A student in the lab, Matt Funston, did this experiment. So he takes macrophages and he puts bacterial digests of gluten on top of the macrophages. And then he looks for secretion of pro-inflammatory cytokines, TNF, and IL-6. And you can see that in the presence of bacterium and gluten, there is high production of both cytokines. Whereas here is the mutants that don't have expression of the proteases, they don't produce high levels of cytokines. So bacteria secrete proteases that liberate peptides, stimulating, sorry, this is, okay. We assume that proteolysis produces biologically active peptides, but experiments were done, and that changed our appreciation of this fact. First of all, it turns out that you can take this supernatant and digestion, and you can try to heat and activate it, and it doesn't work. If it was a protein, a peptide that would stimulate production of cytokines, then it should be inactivated by heat. Moreover, It was not also inactivated by digestion with proteolytic enzyme trypsin, suggesting that this may be not a proteinaceous moiety that stimulates production of these factors. And what happened then is when Matt tried to stimulate TNF production from macrophages that lack toll-like receptor 4 or adaptor MyD88 that are involved in innate signaling, they were not producing TNF at all. So that suggested that because we already know that TOL4 recognizes lipopolysaccharide or LPS of gram-negative bacteria, we thought that LPS could be the factor, and it was true. So if we use polymyxin B either as beads and then or just adding it to the culture, polymyxin B inactivates LPS. So if you inactivate LPS, you remove this stimulation. So the stimulus is not a protein, it's not a peptide, it's lipopolysaccharide associated with gluten. And then what is very interesting, Matt did experiment in which he titrated LPS to the concentration when it by itself, it does not activate TNF production. So this is a black square here. You can edit, it's very little of LPS added. However, so if you, this is the amount of TNF that is produced by digestion of gluten with Enterococcus faecalis, and that is the combination of the two. This is Enterococcus faecalis plus gluten plus non-stimulatory concentration of LPS. And you can see that digestion of gluten by faecalis significantly activates, potentiates LPS compared to just LPS present in gluten without digestion. So the innate arm of immunity could be triggered by small amounts of LPS in the presence of gluten-derived peptides, which amplify the effect. And so what is the connection with type 1 diabetes, if we go back to that disease, is shown by this experiment. So as I told you, if you put germ-free mice on hydrolyzed casein plus gluten diet, here there is no diabetes development. But if you colonize this mice with wild type E. faecalis, diabetes comes back. And if you colonize it with E. faecalis that cannot digest gluten, then nothing happens. There is no diabetes development. So here is the conclusions from my talk, that bacterial proteases can digest gluten, releasing peptides recognized by T cells. They also release peptides that regulate innate responses to components of other bacteria, because E. faecalis is gram-positive bacterium doesn't have LPS, so LPS has to come from other bacteria. These features are likely to contribute to both celiac disease and type 1 diabetes, given that genetic background is permissive in both. And what we were not showing here is that E. faecalis is a prototype, it's not a protagonist. And we know it from collaboration with Eric's lab, with whom we used bioinformatics to identify bacteria in their huge collection of bacteria in the bacterial bank, identify those that may have the similar proteases. And in fact, one of the bacterium that we identified can digest gluten and produce peptides that activate T cells. And I'd like to end here with saying thanks to my collaborators here at the University of Chicago or at the University of Minnesota, Jackson Lab, and in Oslo. Thank you for your attention.
Thank you very much, Sasha, for this great talk. I would like to remind you before moving to the next speaker, I would like to remind everybody that you can ask your question by using the Q&A tool that you'll find at the bottom of your screen. Thank you. So our next speaker is Dr. Valentina Discepolo, that many of you know because she spent many years at the University of Chicago as the first Carlino Fellow in Celiac Disease. So Valentina is now Assistant Professor of Pediatrics in the Department of Translational Medicine Science at the University of Naples in Italy. And she will talk today about the role of viral infection in celiac disease.
Good evening, everybody. Valerie, thank you very much for your introduction. And I would like to thank very much Bana and Ritu for their kind invitation to join this wonderful virtual symposium on celiac disease and autoimmunity. So tonight I'm going to talk to you about the role of viral infections in celiac disease, and I structured this presentation into three main sections. The first part, I'm going to give you an overview of the evidences that we have in support for viral infections, a role for viral infections in celiac disease. A second part, when I'm going to go over some data that we produced in Bana's lab that support the role for viruses in the context of loss of tolerance to gluten. And then in the last part of the talk, I'm going to give you an overview of how we are going to move forward this very interesting field of research. And I have no conflict of interest to disclose. As you've heard already before me, we know that celiac disease develops in genetically susceptible individuals upon ingestion of gluten. And celiac patients typically develop anti-tissue transglutaminase, anti-deamidated gliadin antibodies, and they, at the level of the small intestine, they develop a small intestinal enteropathy featuring intraepithelial lymphocytosis, crypt hyperplasia, and villous atrophy. We have heard as well by Sonia and other people this morning that only a subset of genetically susceptible individuals carrying HLA-DQ2 or DQ8 allele will then develop celiac disease, suggesting that other environmental factors in addition to gluten might play a role in celiac disease development. And among those, the most studied environmental factors have been feeding practices, going from breastfeeding to the gluten introduction in the first years of life, the use of antibiotics, modality of delivery, vaccination, and I'm going to be focusing more on viral infections. One of the first reports that support the role of viral infection in the context of celiac disease dates back to 1984, when Kagnov showed an homology of sequence of the alpha-gliadin with a protein of the adenovirus type 211, and he showed in vitro that antibodies that reacts against this adenoviral protein cross-react with alpha-gliadin, suggesting that viral epitopes might actually be important in promoting an anti-gluten response and thus celiac disease. Since then, many evidences have been produced and in support for viral infection in the context of celiac disease. Not only adeno, but also astrovirus and rotavirus, importantly, have been associated with celiac disease. Antirotavirus antibodies have been shown to be present in over 80% of patients that have a co-occurrence of celiac disease and type 1 diabetes, and they generally appear before the onset of autoantibodies against tissue transglutaminase. Also, a recent study by the TEDdy study group showed that kids vaccinated against rotavirus are protected from developing celiac disease or less at risk to develop later celiac disease. Not only GI infection, but also respiratory infections have been associated with development of celiac disease. Indeed, respiratory syncytial virus, influenza viruses have been associated with celiac. Importantly, the number of infections occurring early in life have been associated. So the higher number of infection, the higher risk of celiac disease. Also, again, respiratory infection in the first 2 years of life, as well as numerous infectious episodes occurring in kids that had larger amount of gluten introduced early in life. Also, genetic studies underlined or displayed that genes, polymorphism associated with genes involved in viral response might play a role in celiac disease. So in summary, more than one viral infection have been associated with celiac disease development later on in life, not only GI, but also respiratory infection. And it seems that the number of infection, the time of infection, and the synergy with the time and the amount of gluten introduced early in life might play a role in the future development of celiac disease. However, it's not that simple, because despite viral infections are very common, HLA-DQ2-DQ8 are present in 40% of the general population, then only a tiny percent of those individuals develop celiac disease. The mechanism through which it has been hypothesized that viral infection may impact the development of celiac disease are multiple. It has been hypothesized that they might promote an increase in intestinal permeability and increase the passage of gluten across the mucosa, that they may promote an inflammatory environment that increases the expression and activation of tissue transglutaminase, thus increasing the generation of immunogenic gluten peptides, and also, as I said at the beginning, molecular mimicry, although this has not been actually proven. I'm going to focus on the mechanisms that support a role for viral infection in impairing tolerance to dietary gluten. And I'm going just to crystallize the complex events that leads to celiac disease in a couple of key steps that lead to development of tissue damage in genetically susceptible individuals. Some of those susceptible hosts upon gluten ingestion develop a gluten-specific T-cell response, as you've heard before me. And this is characterized by the production of anti-tissue transglutaminase antibodies as well. And in clinics, we define these patients as being potential celiac disease when they still maintain a normal intestinal architecture. Some of the patients with these features then go into developing a tissue damage, and they develop full-blown celiac disease characterized by villous atrophy. So again, these key two steps are: one, the loss of tolerance to dietary gluten, and then the acquisition of the activation of intraepithelial lymphocytes that become cells able to destroy the intestinal epithelium and determine tissue damage. And I'm going to focus on this first part. In the absence of intestinal inflammation, gluten is normally taken up by dendritic cells in the lamina propria that, when they have tolerogenic phenotype, they promote more of a regulatory response rather than an inflammatory response at the level of the mesenteric lymph nodes. However, in the context of an inflammatory environment in the lamina propria, inflammatory dendritic cells, when uptake oral antigen like gluten, they might drive an inflammatory rather than a regulatory T-cell response. And work that has been done in Bana's lab have been tried to address the question of what drives this loss of the wall of tolerance and what really triggered this inflammatory phenotype in dendritic cells. And it has been shown that IL-15, actually, in a paper led by Valeria Abadi and in the group of Bana, that IL-15 could be one of the key cytokines that promote this pro-inflammatory dendritic cell phenotype and drive the establishment of loss of tolerance to gluten. However, in that same paper, they suggest that IL-15 does not actually is upregulated in all patients. And we actually went back into patients and looked not only at IL-15 expression in the gut, but also at type 1 interferon expression in the gut. This is a group of cytokines, you've heard about them a lot in the first session this morning, that are produced in response to viral infection. And those cytokines, we use the proxy, which is MX1, that is downstream of these cytokines, and we looked at the expression of IL-15 and MX1 in the small intestinal lamina propria of biopsies of patients. And we were able to identify that there were patients overexpressing IL-15, here represented in red, and those expressing more the type 1 interferon pathway expressed here represented in green, as well as patients expressing both. And in this way, we're able to define two subgroups of celiac disease patients at least, and studied their transcriptional signature in the gut. And we were able to show that we're a shared response in which we saw an upregulation of genes involved in tissue damage, like interferon gamma, non-classical HLA class 1 molecules, and NK receptors, but also specific IL-15 responses and type 1 interferon responses. Among those genes, we identified genes involved in response to viruses, thus providing an evidence in human that there is a viral signature in some of these patients. And so like this, just to schematize that despite they need to assume gluten and to have HLA genes, patients could be different from an immunological standpoint, and probably different environmental factors may trigger distinct immune pathways, but then have the same disease outcome. So focusing on the type 1 interferon upregulation and this subgroup of patients, we wanted to then further investigate if this type 1 interferon upregulation could actually be promoted by a trigger that actually would impair loss of tolerance. And then this is the a work that has been published in 2017, and Roman and Rainey in Bana's lab led this wonderful work, and they studied the role of viral infection in promoting this loss of oral tolerance. To set up the experimental model, they used 2 different strains of reovirus, the same family, T1L and T3DRV. The latter is a reassortant that was genetically modified in order to be able to infect the gut. And the 2 viruses were able to actually infect at similar level the different intestinal tracts. And they were able also to induce neutralizing antibody titers, although T1L at higher level than T3DRV, but they were also both cleared by the host. And in this work, we looked at, and I'm oversimplifying the results to get through this very sophisticated and elegant paper. We looked at the phenotype of the dendritic cells that uptake the oral antigen at the level of the mesenteric lymph node, and we looked also at the T cell conversion upon the viral infection. So mice were infected with either one of these 2 strains of viruses in the context of gluten feeding. And when we looked at the phenotype of the dendritic cells, we showed that T1L promoted an inflammatory phenotype in dendritic cells that were able to uptake oral antigens. And this is shown here on the right, but not T3DRV. When we looked at CD4 T cells, we were able to show that T1L, but not T3DRV, was downregulated the regulatory T cell response, and here we see the FOXP3 positive cells, and also was able to upregulate the pro-inflammatory response, as we can see here represented by the percentage of TBET positive cells at the level of the mesenteric lymph node. And here I show you this graph that Bana earlier showed this modeling to summarize the main finding of this paper, where we actually were able to show that different viral strains that belong to the same family, despite they were both cleared by the host and able to produce protective immunity, they were able, in the context of administration of a dietary antigen, to produce a different immune response against the dietary antigen. In particular, only T1L, but not T3DRV, was able to impair oral tolerance to a dietary antigen, in the specific case OVA, and I'll show you later the relation of this with gluten. And then in particular, the 2 pathways that were responsible for promoting the Th1 response and downregulated the Treg response were not redundant. And in particular, the pathway was involved in the promotion of the Th1 response was IRF1. And how is this relevant for celiac disease? Similar experiments were carried out also in mice that were expressing the humanized HLA-DQ8, and they were fed gluten. So gluten was used as an oral antigen, and similar results were shown. In particular, T1L was able to induce loss of tolerance to gluten in these mice that carried the HLA-DQ8 allele. And again, going back to patients, we showed that celiac disease patients compared to control displayed higher levels of antibodies against reovirus. And in particular, the patients with the highest level of antibodies were the ones who showed higher levels of IRF1 in the small intestinal mucosa. And this is exactly the molecule that we saw to be responsible of the anti-gluten response in our mice. A similar study has been carried out with murine norovirus. I'm not gonna go through these. I'm just gonna point out that IRF1 was a key element. And again, also in this paper has been shown that different strains of the same, of a virus belonging to the same family were able to show different phenotype when we looked at the ability to induce tolerance to an oral antigen. And IRF1 has a key role also in this other experimental setting. So in summary, we show that distinct viral strain were able to impact in a different way the immune system, and also viruses that are normally silent, because reovirus does not infect the human producing intestinal symptoms, was able to impair the tolerance to oral antigens. And then there was a dissociation between the protective immunity, so the ability of the host to clear the virus, and the response to the dietary antigen. And also we show that different type of viruses, so single-stranded and double-stranded RNA virus, could actually promote a similar phenotype in terms of loss of oral tolerance through the same pathway that that is characterized by the upregulation of IRF1 at the level of the gut. So to go back to the schematic that I used earlier, so we show that viruses may impact loss of tolerance to gluten. And so when we go back to our patients, we would then expect that potential celiac disease patients who actually display signs of loss of tolerance to gluten might have an upregulation of viral-induced pathway and type 1 interference. And this is the case. I'm just going to show you that here we see a volcano plot that looks at differential expressed genes between controls and potential celiac disease. And among the upregulated genes, we could clearly identify pathways such as the type 1 interferon pathway and response to viruses. And here we can see representative genes that include STAT1, IRF1, and IRF9 that are upregulated in potential and even more inactive celiac disease patients, suggesting that even in patients that are not celiac, completely full-blown celiac disease, but they are potential, but they already lost tolerance to gluten, we have signs of the type 1 interferon pathway and the viral response. So what remains to be addressed in patients is if there are other viral infections that are associated with loss of tolerance to gluten, if, again, as suggested by epidemiological studies, more than one viral infection actually might be associated with increased risk, and if there are other pathways at the level of the gut and not only IRF1 that might actually be required to promote loss of tolerance. And most importantly, what's the key time window for viral infection to impact loss of tolerance to gluten, especially in relation with first gluten introduction, time of weaning, and time of declining of maternal antibodies. So determining that the first years of life might be key to address this unanswered question. And to try to address this question, and we hypothesize that viral infection that may alter the intestinal immunohomeostasis may occur at the time of weaning, and that may trigger loss of tolerance to gluten and promote celiac disease development in kids that are genetically susceptible. In order to test this hypothesis, we established a collaboration with the Prevent CD group, which is a European group that includes over 13 countries, and they studied the early intervention in terms of low and higher amount of gluten given in early infants that are at risk for celiac disease. So interestingly, this big cohort of patients, they collected serial samples before and after the development of celiac disease, and they were willing to share their samples with us to test this hypothesis. And then we're going to test this hypothesis using one of those newly developed technique that is a very high throughput technique that allows with a little drop of blood to look at antibodies against all the non-human viruses that have been shown to infect humans. And upon correlation of this data on the antibody response with the clinical data, we were able to understand if more viral infection early in life at the time of first gluten introduction will actually be able to promote the onset of celiac disease. And we really think that this study is gonna really open new avenues for designing preventive strategies, such as avoiding gluten introduction at time of weaning when maternal antibodies are declining, or identifying new vaccination strategies to protect infants that are at high genetic risk to develop celiac disease. And we also think that this might be relevant for other autoimmune disorders that are known to be associated and triggered by viral infection. And my last slide is actually a thank you slide. Sorry, I opened the chat because I think I'm over time. I just would like to really thank Bana for her mentorship over all these years at the University of Chicago. All the people in the lab who have been carrying out the experiments and collaborating. I would like to thank Luis, who is one of our key collaborators for all the transcriptional data, and all the people at the University of Chicago Celiac Disease Center, in particular patients and families who actually gave us their samples, the funding and sources and collaborators, and of course, my new family at the University of Naples. Thank you very much.
Thank you.
Oh, I have the bean. Sorry.
Nostalgia. Sense of nostalgia. Thanks, Valentina. So we'll move on to our last speaker now, Dr. Anderson. So Dr. Anderson, and we were in Italy, we're moving further away in Australia now. Dr. Anderson is a gastroenterologist and researcher who made seminal contribution to the field of celiac disease, notably by developing methods enabling to detect peripheral gluten-specific T cells in gluten-challenged individuals. He was the scientific founder of the company Immusanti, and until 2019, he was the chief scientific officer of Immusanti, that developed and tested the first peptide-based therapeutic vaccine for celiac disease. And he's now an honorary fellow at the Wesley Medical Research in Brisbane, Australia. And today, Bob, I don't see him yet, but hopefully I'm going to see him soon, is going to discuss the strategy that can be used to promote tolerance in celiac disease and autoimmune disorders. So.
Thank you, Valerie. Can you see me now?
Perfect. You can share your screen, your slides now.
Great. Yeah, so I really appreciate the chance to talk to you about the work that I've been doing over the last 20 years. I guess I would just like to preface what I'm going to speak about with, here we go. Let me just go full screen. Good. Can you see the talk?
We cannot.
You can?
No.
Oops. Let me just come back. How's that?
Perfect.
Great. Just full screen mode, please. Perfect.
Perfect. Okay. So, yeah, very appreciative for the chance to share with you my thoughts. I really just wanted to preface this talk by saying that over the last 20 years of my involvement in celiac disease, I've been really focused on trying to measure responses to gluten and celiac disease in blood from patients. And the philosophical backdrop to that is really that I believe that you need to try and minimize the artifact in what we measure in many experiments by minimizing the manipulation of blood and what we do to patients. So the data I'm going to share today is all based in patients with celiac disease. It's all based on adults who have established celiac disease generally who've been on a gluten-free diet for a period of years. And I guess the reflections I've had since stepping away from my role in commercial drug development 12 months ago, and really effectively sitting outside academic research, is, I think, a reflection on where I think celiac disease could head and some of the implications of the work that really come out of drug development. So I have a variety of disclosures there related to my work in drug development with various biotechs and farmers who've been interested in developing drugs for celiac disease. And I hold several, I'm the inventor on several patents, but I don't own them. So many of us have been involved in drug development, and many of the drugs that are now in development are targeting the T cell, so the adaptive immune response. So I believe there's an implicit corollary in the title of my talk, which is that tolerance induction is really all about the adaptive immune response. So most drug development for celiac disease is based implicitly on a model of pathogenesis that positions the adaptive immune response to gluten as the underlying cause of celiac disease. And the presumption, therefore, is that if you could remove the adaptive immune response, then gluten would be innocuous for patients with celiac disease. So I think it's time to revise the definition of celiac disease. And when I look at the accepted definition that many of us use, and this is quoting from the Oslo definitions from 2012, celiac disease is defined as a chronic small intestinal immune-mediated enteropathy precipitated by exposure to dietary gluten in genetically predisposed people. And I would propose, given that the work that has emerged, particularly over the last 2 years, is really repositioning celiac disease as a gluten-induced pro-inflammatory recall immune response, with or without typical symptoms of nausea, vomiting, tiredness, bloating, and abdominal pain within hours after gluten ingestion. So it's really saying that it's a systemic response. It's not just the gut. And when we think about what celiac disease is, it appears that it's caused by disease-associated gluten-reactive CD4 T cells, and particularly transglutaminase and gluten-specific B cells and plasma cells. And I would preface that by saying this is in patients with established disease. So given what you've heard in the previous talks today, I think the dendritic cell is critical during the induction phase, but once that adaptive immune response has been established, the preferred antigen presenting cell is likely to be the B cell and perhaps plasma cells based on the work from the solid lab that's been published over the last couple of years. The other key observation that's emerged in the last 2 years is that interleukin-2 release, so interleukin-2 as a classic T cell derived antigen-induced cytokine, interleukin-2 release after gluten ingestion is the hallmark feature of treated celiac disease with or without intestinal injury or elevated transglutaminase serology. And that intestinal injury and or elevated transglutaminase serology are hallmark features of regular exposure to gluten. So we've been living, I think, in a world where we have a definition which works pretty well in people who have regular gluten exposure. But we now have many, many people who are following a gluten-free diet, diagnosed with celiac disease, or maybe not diagnosed with celiac disease, but they still have celiac disease. It doesn't go away because there's no gut injury and the serology is normalized. So we need a definition that really addresses both groups of patients who have celiac disease, but they have different amounts of gluten exposure. So I'll just share with you some of the data that has come out of the development of NextVax2. And we were really trying to work very hard to develop biomarkers in blood that could be collected from patients during clinical trials that would serve as endpoints in therapeutics. And the thing that we came across was that when we fed patients a single bolus of gluten in the form of vital gluten, which I would really highlight is very low in the FODMAP component of wheat flour. So rather than feeding patients wheat flour, which we knew had FODMAPs in it and could really complicate interpretation of symptoms, we fed people vital gluten. And this is about 75–80% protein. There's very little FODMAP in it. So we're trying to assess here the effect of the immune activation in a patient with celiac disease during the 6 hours after they consume gluten. And you can see there in panel A, the overall GI symptoms in a group of 295 patients who all had exactly the same gluten exposure as a single bolus at time 0. That symptoms rose fairly steadily up till 3 hours and then gradually improved. If you look in panel B, when we break out the individual symptoms, it's nausea that's the most prominent. Diarrhea is remarkably minor, and in fact many patients expected to have diarrhea when they had gluten exposure. It was actually nausea and in some cases vomiting, which were the most prominent symptoms. And headache and tiredness also rose as well over that period of time. But as you can see, the symptom complex is actually relatively predictable in these acute gluten challenges in patients with treated celiac disease. And when you look there on the left-hand side, the levels of interleukin-2 rose quite abruptly in people who experienced very severe overall gastrointestinal symptoms. But you could still measure rises in interleukin-2 even in patients who had no digestive symptoms there in the green line, which is slightly obscured there under the gold diamonds. So interleukin-2 release is fairly ubiquitous in patients who have treated celiac disease when they eat gluten. And you can see there on the top right panel, if you look at the histology in a subgroup of these patients, so about 50 patients had biopsies before they had the gluten challenge. And IL-2 release was actually more prominent in the patients who had slightly worse histology, so the group with lysohype depth less than 2. The more healed patients tended to have less interleukin-2 release. And then in the lower right panel, you can see what happened when patients had 2 gluten challenges about 14 months apart. And this shows the interleukin-2 levels in serum 4 hours after eating exactly the same gluten challenge. And there's an extraordinary correlation, very, very strong relationship between the interleukin-2 release and the success of gluten challenges. So this is a very reproducible phenomenon in patients with celiac disease. Amongst those patients who were enrolled in the phase 2 study of NextVax2 from the United States, Australia, and New Zealand, we were able to compare the HLA type in people who entered screening and then had the gluten challenge. And I wanted to share this with you because we were able to show interleukin-2 levels rise in all of these HLA types. And the group that I would really like to point out, and there were very few of them, I would add, were the patients who were HLA DQ7. And you can see there we have a patient who had a very prominent symptomatic response to gluten, who was, for all intents and purposes, the same as the other patients who had celiac disease with classic HLA DQ2.5 or DQ8 or DQ2.2. So IL-2 release after gluten consumption, i.e. reactivation of adaptive immunity to gluten, is ubiquitous. It seems to be present in all patients who have celiac disease diagnosed by standard serology and biopsy with response to gluten. Interleukin-2 is not the only cytokine that rises after gluten consumption. And as you can see there on the left panel, we did quite a large O-link screen of about nearly 190 cytokines in serum from people with celiac disease on a gluten-free diet versus others who were healthy controls. And IL-2 in the confirmatory phase on the right turned out to be by far the most prominent and the most consistent amongst those. But as you can see, IL-17A, some of the chemokines were also prominent. So IL-2 is a marker. It's not the only cytokine that goes up, and it may not be the one that is actually responsible for symptoms, but it's clearly a very strong correlate with the symptom response. This work has been replicated now by 3 separate groups, and this is the most recent one that appears as a paper in Press, Gastroenterology, and the group from the Harvard Associated Hospitals in Boston, along with Takeda, were able to look at a 14-day gluten challenge with blood collected either at 6 days for T cells in blood or 4 hours after beginning the gluten challenge. And the remarkable thing in this work is that the histological changes after eating gluten for 14 days are actually rather inconsistent in terms of response rate, which I've shown you down there in the bottom right. So the villus height and the intraepithelial lymphocyte densities are shown on the left for individual patients. And certainly with a large challenge of 10 grams, most people do develop histological change. But the biomarker that was most consistent was in fact interleukin-2 elevation after 4 hours, which was more consistent than either histological changes after 14 days or using tetramers to stain gluten-reactive T cells after 6 days when they've expanded and present circulating in blood. So this is a very nice demonstration that the IL-2 response with acute activation occurs very, very quickly when people eat gluten. And this is telling us something which I don't think any of us would have really expected, is that the adaptive immune response to gluten is switched on very quickly within just a few hours. And I think it's fair to say that many people have attributed this early acute symptom response and immune activation to direct innate immune effects of gluten, whereas that appears not to be the case. And so you can get a whole panel of biomarkers that come up at different times. It's all part of the adaptive immune response to gluten. The IL-2 response just appears to be the very first one that correlates well with acute symptoms after beginning a gluten exposure if you have celiac disease. But the markers that our current definition of celiac disease are based on are those down the bottom, the ones that occur in biopsies and in blood after weeks or months of gluten exposure. So this is why I think now is the time to revise what our definition is for celiac disease. Further evidence for the T cell involvement in early symptoms came from the work that we did with NextVax2. We injected these peptides I've shown here directly into patients by intradermal or subcutaneous injection in microgram amounts. So these are 3 immunodominant peptides derived from wheat or barley, hordanes. These are really probably the key peptides that drive the adaptive immune response in patients who are HLA-DQ2.5. But I would just point out that each of these 3 peptides have epitopes recognized by antibody, IgG antibodies circulating in patients, particularly the second one, which has the classic immunodominant B cell epitope for gliadin. And when we injected patients with these 3 peptides on one occasion, we found that there was cytokine release. And in fact, it was identical to the gluten-induced cytokine release I just showed you. And we did a discovery phase followed by confirmatory VPLEX assessment of these cytokines. And in the top right, you can see that the 150 microgram injection, intradermal injection of the NextVax2 peptides induced interleukin-2 quite dramatically. So these are systemic cytokine release gluten-induced syndromes that appear to be the same whether you inject peptides that are immunodominant or take gluten. So this really led us to perhaps a new model to understand what's going on in celiac disease and how a drug development program could develop to suppress symptoms and reduce gut damage. And it's really by centering the gluten-reactive CD4 T cell in the middle. So the 5 studies that we did in NextVax2 ended with the Reset CD program that concluded the middle of last year. And I'll just focus on that for a moment. So the NextVax2 peptides are systemically available. They rise after an intradermal or subcutaneous injection very quickly. They peak at about 1 hour after injection and they're cleared after about 6 hours. When we inject those peptides, as I said, we had a cytokine release associated with that, but over repeated dosing, as you can see there, after 3 doses or after 16 doses, there was no immune activation. So repeating the dosing with NextVax2 very effectively reduced the immune activation associated with injecting the peptides. And on the panel on the left, you can see that the symptoms associated with the first dose of NextVax2 really did not occur with the repeat injections over a period of time. So this, as far as I know, is the first demonstration that you can suppress the immune activation and the symptoms associated with gluten epitope exposure. In our phase 2 study, though, we wanted to show that gluten exposure was similarly reduced. We collected blood in a mechanistic study before the patients were exposed to the NextVax2, and then about 12 weeks after, to see if the immune activation associated with the NextVax2 injections was changed. And here you can see, just in summary, that there was a highly significant reduction in the fresh blood cytokine release induced by the NextVax2 peptide. So this is in vitro data showing that the immune activation caused by the NextVax2 peptides is dramatically reduced with treatment. It also reduces the immune activation associated with peptides from gluten that are not in the NextVax2, and that's shown in this column here. So on paper, immunologically, it appeared that NextVax2 treatment was suppressing the immune reactivity in these patients to key epitopes in gluten. The issue was, though, that the primary endpoint and the secondary endpoint were not going to be met during an interim analysis. And this shows you that when there was a double-blind masked gluten challenge associated with a sham gluten challenge, that if we look here, there was no difference in the NextVax2 treated patients in this panel and the interleukin-2 release that occurred after the gluten exposure compared to the placebo treated patients, even though there was a dramatic difference between the sham challenge, which didn't induce IL-2, versus the active gluten challenge here. So the interleukin-2 response to gluten was not changed by NextVax2 treatment. And then I would just add that the symptom response, which was the primary endpoint, was also not changed. And that led to the study being discontinued before it was completed. So the experience, I guess, led to this bullet point list, which is somewhat oversimplified, but key strategies for tolerance induction in celiac disease. The reason we began the NextVax2 development program was because it appeared to be the simplest strategy to potentially induce tolerance to gluten. And there are several other diseases, allergy, which have utilized this approach with promising results. However, it doesn't seem that the tolerance induced by NextVax2 is sufficient to, at the dose levels that we explored, to be able to suppress the immune activation and the symptoms associated with gluten at the dose we chose. The gluten dose we chose was large, so we gave 10 grams of vital gluten, which is probably about half a day's worth of gluten exposure on an unrestricted diet. So you could argue we gave an excessively large amount of gluten. The NextVax2 peptides, I believe, are likely to be presented by B cells rather than necessarily selectively by dendritic cells. And clearly that means that a range of immune tolerance induction mechanisms induced are not going to be those which rely upon dendritic cells, which a number of the nanoparticle approaches to gluten tolerance induction do utilize. So that's, I think, a key difference. The dose optimization that we went through really tried to correlate the level of immune activation induced by the gluten peptides and NextVax2 versus the immune activation induced by gluten, and we thought a dose that was relevant. And we utilized the IL-2 release as a biomarker that would guide that. The other thing that I would stress is that you need a drug that induces tolerance, which is simple to make. And the formulation is stable. All the issues we've heard about with vaccines for COVID, these are issues related to tolerance induction agents for celiac disease and other autoimmune conditions. And ultimately, it's that, are we headed towards a curative immunotherapy that allows patients essentially to walk away without celiac disease on a normal unrestricted diet? Or are we in a world where we're trying to maintain that immune tolerance for as long as we can, but was really reliant upon repeat exposures to that therapeutic agent? Overall, in terms of tolerance to autoimmune disorders, I think we're still really burdened by methods that don't allow us to map relevant T-cell epitopes in human disease. That also impacts on monitoring of those T-cell responses during therapies. This is a major disincentive to biotechs and pharma getting involved in this field. And until that's resolved, I think it's going to be difficult to really attract a lot of players to this field. So we know that whole antigen immunotherapy works for allergy in terms of suppression, symptoms and immune activation. I think that's potentially going to be a pathway that is still attractive in autoimmune disease, but it's very, very likely that we have to address the post-translational modifications to those antigens to allow them to engage with the relevant T cell population and B cell population. The particle delivery approach, I think, is exciting, but I worry that if antigen is exposed on the surface of nanoparticles, they're simply going to be directed to B cells. And is the tolerogenic effect going to be the same if those nanoparticles end up on a B cell as they would on a phagocytic cell, like a dendritic cell, or maybe one of the scavenger, via a scavenger receptor on macrophages? So I think that is a key design issue. The other problem we have is that most of the people enrolling in our trials were about between the ages of 40 and 50 years old. I don't think that's ideal for tolerance induction. And I think our efforts in the end would be better directed towards children who have a more malleable immune response. So we may not be able to show tolerance induction simply because the immune response is so well established. It's very hard to reverse it. And it's really whether symptoms as the primary endpoint may cause an unwarranted number of late-stage drug failures. I'm not sure that the patient-reported outcome tools we have are optimal. And given the work that I just shared with you on the symptoms caused by gluten, these are not the symptoms that were previously attributed to celiac disease and gluten exposure. So I think we need to have a careful think about the PROs that we use. So I'll finish there, but basically I'm very grateful to everyone who was involved in all the NextVax2 development, the patients, and we had a very strong relationship with the Walter and Eliza Hall Institute, Jason Tydin, who helped out with a number of the studies that I shared with you in the talk. But thank you very much.
Thank you, Bob, for your talk. So it's a wrap for this session. I'd like to thank all the speakers who are going to answer your question now. And I would like to thank again the Celiac Disease Foundation, who was a sponsor for our session. So thank you. So yes, Bob, maybe you can stop sharing.
Sharing.
I'm gonna ask everybody to switch on their video. So we're gonna start with the questions. We actually can start with the question for Dr. Anderson, because we just finished without. So there's a question for you, like, in your opinion, which patient population, symptomatic versus asymptomatic, those with incomplete healing or those wishing to ingest gluten or to avoid gluten cross contact, should be studied for non-dietary therapies?
So we had lots of discussions with Bana about this issue related to the damage in the gut and whether patients with damage in the gut would be very hard to tolerize because of the innate immune activation corrupting the dendritic cells. The difficulty really in that discussion is that we discovered in our patients enrolled in trials who appeared to be well controlled with normal serology, adhering to a gluten-free diet, 2/3 of them turned out to have villus atrophy when careful quantitative histology was performed in Finland by G-Lab, Markumaki. So you can't really escape the gut damage. It's going to be there in almost all the patients you study. And limiting trials to people with no damage will almost make it impossible to enroll patients in trials. The question of symptoms is, I think, a bit misleading, because I suspect that many of the symptoms that people complain of with celiac disease on a gluten-free diet are in fact irritable bowel syndrome driven by FODMAPs. I think there are definitely symptoms related to gluten exposure, as I shared with you, but many patients don't recognize them necessarily as the key symptoms of gluten exposure. So it comes back to histology and symptoms, as I said, that you may want to use biomarkers like IL-2 and cytokine release in blood after gluten exposure to explore whether your tolerizing therapy is successful or not. And I think driving the biomarkers early will give you a good sense of whether your drug is going to be successful.
Valerie,
Yeah.
you're on mute.
You're on mute.
Another question for you, like that we keep with the same theme. So do you think that IL-2 could actually induce Treg cells and suppress symptoms if given enough time, and perhaps would overlapping gradual introduction of gluten orally have helped tolerance induction? It's a question of dose, and there's another question related to the dose. Do you think that the dose and it's the dose and timing, basically, how that could influence actually the outcome of tolerance induction?
Honestly, I think it makes a lot of sense to introduce the gluten slowly. We find clinically, and many of you may have found this too, that if you do gradually increase the amount of gluten patients eat, you don't get very much in the way of symptoms early on, and you can gradually increase the dose. The problem in the end, though, is that the gut damage occurs. So there's a dissociation between the gut damage and the symptoms when you're doing that slow introduction. We showed and published that gradual increase in the dose of NextVax2 could allow us to deliver very large doses of NextVax2 peptides by injection without any symptoms. So this idea that you can gradually increase exposure without immune activation and cause a non-responsiveness, I think is well-founded. And that I like the idea, partly because you don't lose volunteers from your trials if they're not experiencing bad symptoms. And that would be a great disappointment if you suddenly lost all of your patients from both active and placebo treatment in a trial. It would be disastrous.
And do you think actually the route of administration of your peptide also influence the outcome? That using the subcutaneous route of administration actually impede the activity of the peptide or the uptake of the peptide, and that decrease actually the magnitude of the response one could expect?
I think, well, going back to our studies, we clearly showed that gradual introduction of the NextVax2 allowed us to deliver very large doses of Nixvax2. What I don't know is whether the final maintenance dose of Nixvax2 that we administered was the optimal dose. I mean, it's hard to know that, and that may have been one of the factors that influenced the outcome of the trial. So maybe if we had gone up to 10 times higher dose of Nixvax2, it's possible that it could have dealt with the dose of gluten. The other way, though, I think, and this is really key, is to view the role of the therapy. So are you trying to improve the health of patients who remain on a gluten-free diet, who are exposed to relatively small amounts of gluten? It's possible the Nixvax2 at the dose we administered could still have been very helpful to patients, because we actually did see borderline significant improvements in the histology of the gut. So it appeared to be doing something beneficial, but it may have only been effective with relatively low gluten exposure for the dose that we were giving.
Okay, thank you. So we have a question for Dr. Shavonsky. So the first question we had is based on the case of a patient who developed both type 1 diabetes and celiac disease, and actually didn't display a classical symptom of celiac, but instead had the drop of blood sugar level after eating gluten, and there's this association between blood sugar and insulin dosage and reaction after eating gluten-containing food. And the question is then, so have you seen this type of connection in general between type 1 diabetes and celiac disease? And do you see a link between gluten and insulin taken together, and how this could affect food absorption? Sasha? That's a tricky one.
That's a tricky one. The short answer, I have no idea. The longer answer is definitely that needs to be investigated more properly.
Okay, there's another question that I lost right now because all the questions came at the end. Okay, so there's a question related to the microbiota. So your work remind of the work by Dr. Verdu and Caminero, find that small bowel, Pseudomonas aeruginosa, both could digest gluten, into immunoreactive peptide. And so the question is basically, have you looked at this bacteria that has been found in other studies looking at celiac in your lab, in your model?
Yes, we're certainly aware of this work. But we in my lab, we don't work with Pseudomonas. We are trying to work with commensals that we isolate from the mice that we deal with in our colony, and to try to connect their ability to digest gluten to development of type 1 diabetes. In parallel, we are investigating huge panels in collaboration with many people, including Eric, bacterial panels, trying to find similar properties of bacteria in humans. Pseudomonas aeruginosa sort of wasn't on our radar, but the point is anyway is that many bacteria can have proteases like elastase in case of aeruginosa. And some of them can digest gluten to activated peptides, and some may even digest it to amino acids, removing peptides, activating peptides. So that's an area that we're very interested in. There is a competition at the bacterial level for that.
Thank you.
Plus, there is also competition with digestive enzymes, and that we don't understand at all yet.
Thanks. And another question is to understand the role of LPS in your in vivo model. Have you looked at the level of LPS after gluten plus hydrolyzed casein administration?
We haven't looked at the levels of LPS after administration, but we definitely looked at the levels of LPS before. And moreover, we went to a supermarket and bought several different powders and several different gluten products, and they all contained LPS by LAL assay, which is very sensitive. The concentrations are detectable, but they are not directly activating. So you need to digest with protease, and then it activates the LPS activation.
Can I just ask a question related to that?
Sure.
Did you do the same experiment digesting the gluten peptides in that context and seeing if when you add LPS and you digest gluten peptides, if you still see the same effect?
Well, the system we're using is very rectified. It's hybridoma T cell that responds to peptide. So I don't anticipate any effect of anything on that of LPS in particular. Because
I was thinking about the TNF induction in the experiment where
yeah, that's a good point. No, that hasn't been done yet.
Okay, thanks, Sasha. There's also a question for Dr. Discepolo. So I know she answered, but I think a couple of them actually should be said out loud because it's of interest.
I can summarize all the ones that were directed to the viral part, I think. So most of them relate to infections that occur more later in life. So infections that occur in adults. And so I received a question that actually asks about this point. So to kind of give a broader answer, there are a lot of infections, not only viral infections, that can create something that is called post-infectious IBS, which is an irritable bowel syndrome that is in response to any sort of infection. And this is not very well studied. So it might very well be that infections of different sorts might create an environment in the gut that is kind of an inflammatory environment, or they create tissue damage, and some of those might actually have an impact of later development of celiac disease. But we think that the mechanism of this is different from the early infections that might promote loss of tolerance. We don't know, and there's not very many studies in adults showing that once you have tolerance, then you can lose tolerance. Although there are cases of celiac disease where you can develop the disease later in life, suggesting that you can also lose tolerance to a gluten after you develop you had tolerance to it. But in terms of the association with infections that are related to this loss of tolerance, those have not, they're not evidences in adults. I think this pretty much summarizes that. There have been specific questions about mycoplasma and HPV, and there's no data on that. And
parasitic infection, the same, or the more exotic infection, let's say.
Yeah, there's very few reports, just anecdotal cases of celiac disease in response to Campylobacter jejuni, Giardia lamblia, but very case reports that do not really allow us to make any general conclusion on that.
Thank you. And so I have a question for Dr. Palmer, because I think it's a very exciting new line of research to be able to pinpoint the metabolite produced by given bacterial species. And so it's more long-term question. So how do you intend then to test the beneficial effect of each species? And do you think that you have to test it, taking into account the fact that bacterial are living in community, and then you probably have layers of communication and crosstalk between different bacterial species that have to be maintained?
Yeah, I think that's these are the great challenges that the field is confronting right now, is how to assemble these. We and a number of other groups, of course, as well, are starting to study this, and they're complex interactions. And I think as we study things from the bottom up, there are, of course, other groups like Marin's group at the University of Chicago studying things from the top down, that it's going to be a mix of machine learning algorithms and experimentation in cultures, also looking in germ-free mice that are colonized with these subsets that are going to have to be done before these things are going to be able to move into the clinic.
Yeah, definitely. Thank you. So I think we're perfectly on time, actually. A message I need, so I'm going to leave Bana say the final words to culture this meeting. Thank you again to all the speakers for this amazing day. Thank you.
So, yes, I'm going to share my screen for the thank you slide. So first of all, a big thanks to all the speakers and moderators. I think it has been a great day of discussion and interaction, and we all felt that it was very important to think about celiac disease beyond celiac disease and in the context of autoimmune disorders. And we are also extremely grateful to all our colleagues in working on type 1 diabetes at the University of Chicago. And I think this day also illustrates the great collaboration that exists here at this university. So special thank you to the sponsors, Brooke and Duncan McLean, Karen and Eric Simon, the Celiac Foundation, Brian and Kat Miller, Nada Milakovic and Michael Gray, and Dana and Moore Goldberg. Really many thanks. And we have a wonderful Celiac Council, and not only are they helping us by providing the means to do what we are doing, but they give us amazing input, and we have wonderful exchanges with them. I really would like to give a very special thank you to Karen Wu and Peggy Hasenhauer. They both work for the Kovler Diabetes Center, but they have become wonderful collaborators for the Celiac Center, and they're working intimately with us. And this day would have not happened without their help and with all the amazing work that Karen Wu did. I want also to give a special thank you to Brad Jutras and Mary Gibson. So we have a very special development office because they don't only want to help us to get in contact with patients who are interested in donating for particular diseases, but they also foster interaction between people at the University of Chicago. And then many thanks to Lisa Gosubowski, Monica Knight, Lindsay Mitchell. I mean, again, without them, they made they did an amazing work in making everything work so smoothly today. And we are really extremely grateful to all your work. And we are also very grateful to Tom Weber that really helped us with CME and accreditation. So thank you very much to all of you. Thank you for those in Italy and other places who stayed up very late. Thank you, Bob, for talking to us from Australia. And again, thank you to all the attendees and to all people who have participated today. And I hope to see you soon at another time at the Celiac Center. And I just want to finish also by thanking Ritu Verma, who is a really wonderful co-director for the Celiac Center. And it has been really wonderful to work with her since she joined the University of Chicago. So thank you all. And I will stop sharing. And
thank you, Bana.
Bye-bye.
Bye, everybody.
Bye.